Good morning. First, I'd like to thank Emmett for inviting me to speak to you today. I'll be talking to you about OCTA biomarkers in neovascular AMD. Primary endpoints in retina clinical trials are usually functional. In particular, visual acuity has been used in many trials. However, the FDA does also require structural endpoints. Ideally, these structural endpoints should also be able to predict the functional response. They also help to prove treatment effects, and ideally, they should be reproducible and quantifiable. OCT is a precise and quantifiable imaging technique that we're all familiar with, and it's become critical to ophthalmology practice. And it's also become a very important source of biomarkers in clinical trials, particularly in wet AMD, as it can provide both quantitative and qualitative data. We've become very familiar with graphs like this, where a decrease in thickness can be a surrogate for visual acuity improvement in wet AMD and in other retina diseases. As the visual acuity improves, the thickness on OCT goes down. There are also a number of qualitative biomarkers that have been used in trials. For example, fluid in the retina has been used as retreatment criteria in PRN arms. Retinal pigment epithelial detachment and subretinal hyperreflective material have been used as inclusion and exclusion criteria in trials. However, there are some limitations of the OCT biomarkers. For example, thickness may not necessarily predict function. If by chance the retina is abnormally thin, then it may not necessarily predict visual acuity. Also, OCT appearance may not necessarily correlate with disease activity. And perhaps most importantly, we're unable to distinguish the neovascular tissue on these images. Does the shrim represent the neovascular tissue? Does the pigment epithelial detachment represent neovascular tissue? Well, now we have OCT angiography, which can really help define where the neovascular tissue is on these images. The principle of OCTA is that you basically take a number of OCT images of a select piece of tissue, and then you detect movement between the images. And really, you're looking at red blood cell movement within blood vessels. So if you see lots of movement, there's high flow through the blood vessels and you get high signal. If there's little or no movement, then you get no signal. The benefits of OCTA are that it's rapid, it's non-invasive, there's no dye leakage, which helps to look at the neovascular tissue more clearly, and it's also in 3 dimensions. And so the 3-dimensional view really helps you to look at specific layers of the structure of the retina and also look at the blood vessels in particular layers. Here's an example of on the bottom you can see a normal outer retina normally doesn't have any blood flow. And this can be really helpful in wet AMD because if you segment out that area in patients with CNV, you can clearly see the neovascular membranes. There are a number of potential biomarkers that might be able to be used in wet AMD using OCTA. You can look at the presence or absence of CNV, the morphology, and the different subtypes of neovascular tissue. And you can also measure the CNV tissue. We did a study where we looked at 45 eyes with wet AMD. Half were clinically active and half were clinically inactive based on some clinical and imaging criteria. We also did OCTA on these eyes, and what we found is that in eyes with detectable CNV on OCTA, most were clinically active, and in the eyes with no CNV on OCTA, most were clinically inactive. In other words, the presence of CNV on OCTA correlated with clinical activity, and the absence correlated with inactivity. However, there are some eyes like this one where the disease is clinically inactive, and yet you still see neovascular tissue. And so there may be other characteristics that we can look at to further define disease activity. There are different morphologies of blood vessels, and there have been some studies trying to correlate disease activity with the morphology of the CNV. And also the morphology can change with treatment, and this may be an interesting marker to look at. We can also look at the specific location of the neovascular tissue. Is it under the RPE, above the RPE, or in the retina? Here's an example of a mixed type CNV, and you can look at the specific components and the size and shape of the classic component or the occult component. Quantitatively, you can also measure the size of the CNV. You can either measure area or linear dimension or even volume. And you can look at the size response to different drugs. And you can even measure the individual components of CNV. This case is an interesting example because you can clearly see the size and shape of the type 1 component of the CNV, whereas on the FA, the dye leakage actually obscures the visualization of that neovascular tissue. The limitations of OCTA and wet AMD are that these biomarkers haven't been yet validated. The measurement methods still need to be standardized. The measurements need to be shown to be reproducible, and they should vary predictably with disease activity. But with the future incorporation of this technology in trials, I think we'll get a lot of this information soon. So in summary, OCT has provided us important biomarkers in retinal trials in wet AMD. OCTA allows us to better visualize the neovascular membranes, and different characteristics of these membranes on OCTA may become useful biomarkers in future trials. But these biomarkers still do have to be validated. Thanks for your time.